Our next speaker is Yael, who I've already mentioned, is our technical director and manages the immunoassay group. And so she's going to give you a more in-detail look at Luminex and other immunoassay platforms. And you can put on this mic. I'll get you plugged in. There, and this is a clip somewhere.
Oh, you've connected.
Oh, that's nice.
Oh, we got a problem here. Let's see.
There we go.
It's okay.
Good.
Okay. So hello everyone, I'm Yael, and I'll be trying to compare apples and oranges, and I hope I try to make some sense for you on this level if we are able even to compare apples and oranges in terms of Luminex and MSD data. The stage is such that we'll be looking at proteins, soluble proteins, and measuring soluble proteins on different platforms. And as Holden mentioned, what I want to do, just to set the stage, is to emphasize that advances in technology allowed us to move from single-plex measurements of ELISAs towards a more profiling kind of view, profiling the different number of soluble proteins that were of interest. And the stage is such that what we're intending to do is try to profile and get a view of distinguishing healthy from disease, as Holden mentioned, also looking at pre and post treatment of different treatments for different diseases, and also looking at different milieus. This allows us to get a steady state level, or kind of fluid-based fluids in the body. And so in the HMC, we use actually two major platforms, and that is the Luminex assay, which Holden started to describe previously, and the mesoscale platform, which actually is an electrochemiluminescence platform. Both of them are type of multiplex ELISAs. So what we've done, we've moved from single measurements to multiplex measuring of different biomarkers in different fluids, as mentioned. So I'll start just with a quick overview again of the Luminex. And basically what we're doing is we have a range. Each sample is actually contains multiple thousands of different beads that have these spectral addresses. This allows us to build an ELISA on a bead, where this antibody is captured to one of these types of beads with a spectral address. Sample is added, attaches. The secondary antibody then attaches. The fluorescent reporter is then later intersected. So when it passes through the flow cytometer, we're able to quantify the amount of fluorescence bound to the secondary antibody. And we can also tell which bead it is, what is the spectral address of this bead, indicating TNF alpha, IL-1 alpha, etc. So the electrochemiluminescence platform for MSD is actually pre-patterned arrays. So what we receive from the company is pre-patterned capture of up to 10 analytes in each well, segregated at the bottom of the well. Again, we're building this ELISA at the bottom of the well. We're activating the signal, amplifying the signal using chemicals to emit a light signal. The light that is emitted is captured by a CCD camera and later translated into intensity numbers. So some comparison for Luminex and MSD. In general, I would say that the advantage of MSD is mainly the fact that you can read a plate in a few minutes. You're limited with the amount of analytes that you can measure for each well, and that has not changed in recent years. But it's very specific and may be pushing better towards the picogram level. However, the Luminex is very powerful in the fact that we have moved recently from 51 to 63-plex in human, as Holden mentioned as well, and also a 38-plex in mice. And this has a very big advantage because we're trying to look at complex profiles. Both of them use a very minimal amount of sample, which is a very big advancement compared to what we used to have with ELISAs, where you need a lot of fluid in order to go through the different testings that you would like to do. This is a fluorescent readout. This is a light readout, light intensity readout, and so that's where they differ. And this is just to show you that really this profile is very comprehensive. But I would like to point out that actually, as I mentioned before, since we're looking at a kind of, you can think of the fluids that we're measuring as a kind of equilibrium, steady state level of the tissues or organs that they're representing. So it can give us a profile, but in depth of which cell it's coming from, you probably will be difficult to make a good guess. We hope to advance there, but actually there are other technologies that specifically address that. So what type of fluids do we measure? And so the major ones are cell culture supinatants, homogenates, and many others that you can see here on this list. And actually anything that you can liquidify, you could measure, even cells, but there's no point in doing that specifically. So all the different types of fluids that you would like to measure, you can try and measure. Some that are not mentioned here are classical tears and saliva, et cetera, et cetera. The major one being, I think, that we do, especially in humans and in mice, is serum and plasma samples. These form a very challenging measurement because these are very complex matrices, and as such, it's a challenge to try and profile all of them without hitting problems of nonspecific interactions and inhibitory factors that are in these milieus. So one of the things that I would like to have you take home as a message here is that you really need to make sure, in order to do a good job in profiling, you really want to make sure that your protocols in place are good protocols, that everyone follows the same protocol, and that you design it very well in terms of the type of collection, whether serum, plasma, etc. Another very important issue is storage conditions. It really is important to make sure that you store your samples and actually aliquot your samples up front and use an unthawed aliquot to get good measurements. Otherwise, you'll have compromised measurements. The other issue, which I'll elaborate in the next few slides, is actually the matrix effect. And in general, you would like to include, that's another take-home message for you guys, is you would always try to include a control sample that you repeatedly use over and over again in each of your runs or testings that you perform. This will allow you actually to compare your runs over time and pick up any issues that have technical limitations. So what I'm showing here is actually different collection tubes that are available indicating serum, heparin plasma, and EDTA plasma. And these complex matrices are really something. So before I move on, what I wanted to point out is these form a very big challenge. So can you exchange or can you interchange plasma for serum, different plasma samples, etc.? And the short answer is no, and I would like to show some data to actually convince you. So here what we're taking is we're taking one subject's serum sample and plasma sample. If it's concordant, you expect a one-to-one correlation, and you can see that it's true for a major part of them, but there are definitely some that are higher in plasma or higher in serum. And so you have to look at this with caution, but more so when we look at a number of samples. So you have 8 subjects here comparing. Higher in plasma is going to be on the blue shades. Higher in serum is going to be on the red shades. This is a heat map. And I know that you can't see, but what you can see here, and that is the important part, is that not only are there some that are higher in plasma versus higher in serum, but also what you can see is that actually every individual has their own specific pattern, and that also ties into some of what Holden mentioned with Jake Glanville, which is currently working on the profiling of these samples. So you can see that for this individual, for instance, this cytokine may be higher in serum. However, in this subject, it's higher in plasma. So it really is, the pattern is really complex, and there's no one fits all. So if I wanted to come up with a factor and tell you leptin is always higher in serum than plasma. So that, for instance, does make some sense, but in certain cases. But for most of the cytokines, it really is subject dependent. So you can't just say overall that for this cytokine, CSF, it's always higher in serum or higher in plasma. Yes?
That's a really dumb question. What's the difference between serum and plasma?
Yeah, so I thought I had that one there. Didn't I have that? Yes. So in serum, what you're doing, you're activating the clotting cascade, and you're removing the clot.
Okay.
Hence, you don't have the clotting factors. Whereas in plasma, you're just inhibiting them using heparin or EDTA. They're still present. So that's the major difference between them. Otherwise, they're very similar. But as you can see, the patterns are completely different. And thanks for pointing that out.
So you're inferring that the ones that some of these are pulled down with the clot?
You know, it really depends. It's not a simple question or an answer, because it's true that you're pulling down a clot with some of the factors, but I don't think that's the only reason for the differences between the individuals. The differences between the individuals is driven by the different profile of each individual. So some may have higher inhibitory factors for a certain cytokine while others do not. And that's still to be figured out. We still don't have a clear answer on who has what and what is the significance of all of these. We hope to find that. So we are seeing some immunotypes, what Jake Glanville calls immunotypes, and it still has to be correlated with some differences, actual differences. It's not clear, but some behave a certain way, others behave a different way, with different patterns of cytokines that may be elevated or reduced. It's not clear how that exactly connects to why they're elevated or why they're reduced, and if it's good or if it's bad. So the range of good is kind of broad. Healthy individuals among us can have up to a 10-fold difference in certain cytokines, and it's still considered normal, as far as we can tell. So another important factor when we move to these complex matrices is, okay, we're looking at, we try to take the signal that we're measuring, whether it's light intensity or whether it's, and I'm concentrating now only on Luminex data for these few slides, but we're trying to measure the median fluorescence intensity and come up with a correlation to concentration of picogram per mil. That actually stems from the fact that we were used to looking at single ELISAs, trying to quantify the best we can, having a cutoff to say, this is normal, this is not. Now, when we move to multiplex profiling, that is a little bit of a challenge. And one of the reasons is, as you can see, the blue line represents all of the cytokines in the standard. So this is going to be standard number 6, which is around 2,500 picograms. And you can see these are the MFI levels for that standard in a buffer. When we spike the same standard into samples, this is what we get. So this is the same standard in actual serum samples. So look at the reduced level for some of the cytokines. And I ask you to ignore the ones that are higher, because those are endogenous. Those are cases where endogenous cytokines actually have higher levels than we have in our standard. Okay, so we cannot measure them. It just appears much higher than the actual standard. But you can see that's true for serum, and it's also true for plasma. These are different individuals spiked in with the same concentration of standard, showing that the signal is reduced. So what does it tell us? What it tells us is actually that we're underestimating. If we were to say, based on our standard curve, that this is the 2,500 mark, this is the level that we would say, this is 2,500 picogram, what we actually see is that we're underestimating, because when we reach the 10,000 MFI, that's the 2,500 picogram level, the real picogram level in the sample, in these samples. So when we use these standard curves, it's an approximation. It's not an exact 2 picograms, 5 picograms, 20 picograms, or whatever number we're trying to quantify. That is a challenge because it's different from the way we were thinking up till now in terms of trying to get a valid or a proof for a certain level and have a cutoff and say, this is normal, this is not. And that relates to the type of matrix that we're looking at. So another way to look at this, because you can't appreciate the actual differences here, I have to log scale these in order to see all of them presented here. Otherwise, if I would not log scale, you would only see the high ones. Okay? So if we go again to this type of heat map, where we're looking at both serum and plasma, and we're looking at percent recovery. Again, I know that you can't read the percentages, and it's not important. Again, what is important is the overall profile. And the overall profile, again, is in general, percent recovery is really not 100%. It's 100% or more for a very minor subset, which are the ones that we can't actually really measure because they're higher in the samples than in our standard curve. For all the others, you have a complex range, again, varying between individuals and varying between serum and plasma, however, displaying the same phenomena in both serum and plasma. It might be that there's a little more, it's also dependent. There are some differences between serum and plasma, but those are not very significant. In both cases, we're underestimating. We're underestimating the quantity of any given target that we're looking at. One of the major differences between them, which I just want to point out, and I don't know if I have it here on my slide. No, it's already out of my slide. But one of the differences is that we see that, so before I go there, just one of the differences that we do see is I did not mention, but we include 4 control beats. So in addition to the 51 or the 63 plex, we always have additional 4 control beats that help us examine and qualify our results and actually test our different steps of the assay to make sure we included everything, all the steps, the correct amounts, etc. That will be elaborated on in the demo. I can show you how I use those beads and how we use those beads actually to qualify or disqualify certain wells. However, one of the differences that we did note is that in serum, the nonspecific binding is a little higher than in plasma. And that I'll be showing you later. So one question that came up, okay, is this happening also in the mesoscale platform? Because we're looking at a very complex multitude of biomarkers. Does this happen also in the mesoscale platform? So as you can see presented here, and I'm only presenting the 9 common ones because we're limited, right? We can only look at these 9. You can see that in general, there's no matrix effect or almost no matrix effect in MSD with the exception of IL-6. So there is some effect for IL-6, but it's minor. The other question that we wanted to answer is there a difference between using polystyrene beads and the now magnetic beads that are mostly where all the field is moving towards the use of magnetic beads for many reasons, one of them being that the CVs are better, coefficient of variation, and there's less nonspecific interactions. However, as you can see, these are kits from different vendors, different types of kits. In both of them, you can see this effect. So it happens in both polystyrene, magnetic, and to a lesser extent in MSD. We think it might have to do with the fact that physically the capture antibody, the first capture antibody, is physically separated in the plate. And that kind of maybe makes these interactions a little less significant. I think also that in general, the fact that we're only dealing with 9 at a time, we have less nonspecific interactions going on in that. In this platform, but it's also much less comprehensive in terms of our measurements. Another important one to emphasize, another important issue to emphasize is that we realized early on when we were looking for these effects that if we dilute the sample, we kind of reduce the matrix effect. So we're reducing some of these interactions, inhibitory factors that may affect our measurements. So the driving force to dilute the sample was very large. However, again, going back to the quantifying part of it, what do? Do we correct for this dilution when we dilute threefold, or do we not correct for it? And the typical answer is we do not correct for that. And the reason we do not correct for these ones is because what we've noted is you would expect that a threefold dilution would result, if the assay is completely linear in those ranges, at a threefold reduction in MFI. This is actually not the case. You can see what I'm doing here is we're comparing subject 1 neat versus diluted, and we have a heat map that shows the deviation from the expected fold change of 3. And you can see that almost none of them are 3. There's only one, IP10, that actually has a higher recovery in the diluted, meaning that we're actually getting rid of some kind of inhibitory effect. But in general, when you dilute threefold, you at best get twofold dilution of twofold recovery of MFI and not a threefold reduction in MFI. So again, this is telling us that the assay is a biological matrix. It's not math. It does not behave in the expected way. Therefore, just correcting by assuming that a threefold dilution is going to result in a threefold more quantitation is not the way that we want to go. It is not correct biologically. So I want to summarize a little bit, and just to say what I presented so far is we really have the dilution that drives a nonlinear pattern, and that is true for both serum and plasma. There are a few exceptions, like for IP10 and resistin. IP10 I've shown you. Then again, plasma and serum are equally nonlinear, so it's not one of them, or it's both of them. Serum MFIs in general are higher in serum than in plasma, and that I haven't shown you, but I will be showing you hopefully in the next slide. I think I have some time for that, yes. So serum and plasma levels are subject dependent, and the cytokines are deviant from the expected ratio of 1. So if you do want to compare, you have to consider that. You can't just interchange a serum sample for a plasma sample because you're missing one time point for a certain subject. It really is not comparable. Matrix affects the detection of the standards, and that leads us to the caution in quantification. So we think it's a relative quantification. It's an approximation. It's not an exact number. It really depends. It'll vary between vendors. It will vary between kits. It will vary between lots. And I'm hoping to show you some of the ways that we can deal with these issues. So again, how do we present the Luminex data? Do we look at MFI? Yes, I say yes. Do we look at picogram? I say no. And again, I'll show you more about check speeds and how to normalize in the demo. But just to give you a reason, again, to emphasize, if you look at this standard curve, you can see that towards the lower end of the curve, really a relatively small change in MFI will lead to a much larger change in what I'm quantifying for the picogram per mil. So what will happen is, let's say I have a control sample. One day I measure it at 20. The next day it will be 25. It can be the difference between 1 to 2 or 2 to 10 in some cases, while it's the same number for the MFI. So you're introducing noise. You're introducing an unreliable measurement instead of looking at the MFI. So it's much better to be looking at the MFI because it's less resistant to this behavior due to the correction. And here is the slide that you can really appreciate that and this is true for many different samples. So in this subset of samples, I have one subject, 3 time points of serum and of the plasma. And the reason I'm showing you this slide is there are a number of reasons. One is that you can appreciate the levels of the serum versus the levels of the plasma. The other one is to show you that, I'm not sure that you can see here, but you'll see better in the next slide, which checks for nonspecific binding in the sample, is much lower in plasma than in serum. So the other reason that I'm showing you this set of data, which is very limited, it's only one subject comparing the serum and the plasma 3 time points, pre and post surgery, baseline 1 hour, 24 hours. And what you can see is what I've done, instead of presenting picogram per mil or presenting even the MFI, I'm moving away from the MFI. I'm using an approach called ratio, which is a very simple, straightforward, intuitive method, comparing, dividing actually time point 2 over time point 1, time point 3 over time point 1, and receiving these nice graphs which show us actually, so just to show you, this is the same subject, serum, plasma and serum for the 3 time points for IL-10 and PAI-1, serpin E. And what you can see here is that both behave the same in plasma, whereas in serum, it's non-detectable. The change for PAI-1 is non-detectable in serum. It is detectable in plasma. Same here. Another subject, plasma and serum, you can see a change that is detected in the plasma, not detected in the serum. Another subject, these are the 3 different subjects, plasma, serum, the 2 cytokines, and you have another set here. So it's a complex pattern, and what we do is we can create all these graphs and look at them individually. It's more telling than the previous slide, where you can't really tell apart those intricate little differences among the time points in the sample. This method is very useful. It allows you to actually bypass any difference between batches, plates, et cetera, et cetera. And again, I will try and elaborate on that in the demo. Another, last one, do I have another? Okay. So another issue that I would want to show you is okay, we know that when we run samples today, we have a certain batch or lot of kits that we have. A year from now, we probably will not have that same lot. And there are going to be differences between these lots. How do we compare data across these different lots and batches? And in the next slide, we're looking at a different set of 8 individuals from the flu studies, actually run on 3 different batches of kits that we had in the lab. None of them are already relevant anymore because we don't have them any longer. But what we did was we wanted to have a way to compare them. This is a heat map for these cytokines. And you can see that there's a block here, a block here, and a block here. Very obvious block. Even though it's the same individual run in different batches, their profile is a little different depending on the batch. So how do we normalize that? One of the ways that we found to normalize this data set is to use what is called a z-score normalization. Here I did not use the log2 in addition, but you can either add a log2 before you z-score or z-score. What is the z-score? The z-score actually is, instead of using the standards, we're forcing all of the data in between plus and minus 3.5, having the median of those specific samples set at 0. So each sample will be represented as either above that median or below that median, looking at all those samples in the plate, excluding standards and excluding any samples that are not related to the subset that you have. One caution on this approach is that you have to consider, and that goes to another issue that I'll elaborate in the demo, you have to design your plate very carefully. You want to always include, if possible, equal numbers of controls and disease, or controls and treatments, et cetera, et cetera. So you want to have the same distribution of samples across plates, across batches, et cetera, before you can normalize this way. What you can see is if I compare this pattern to the z-score pattern after, you can see still that subject number 02 is high, but is much more comparable, et cetera, et cetera. If we go one by one, they're not identical, but we've gotten rid of the batch effect. So you no longer can see a clear batch association for each set of data that you've created. And that's what's nice about this type of z-score normalization. Okay, these are things that I will be elaborating more in the demo on other, we've touched upon some of these, and as well as the different Luminex variables, which I've also touched upon, standards, use of controls, etc. And I would like to thank you, and I hope I didn't juice you up too much, and I would like to thank everyone, especially Iris, Holden, Cindy, Mike, Glenn, all the members of HIMC, which I just didn't include because I wouldn't have enough space. I'd like to thank the Mirai Bio people, the people that we worked with to get kits, subsets of the data. I haven't presented any data for Heather and Suki, but I will try and show you some of the data comparing different technologies in the method that I've shown you before. Cytoanalytics, which allows me to export all of the data in one nice big whole set instead of working on an Excel sheet for each of the reports. Mark Davis, Leo Hansman, which the samples that I presented to you were done in collaboration with Mark Davis's lab. Martin Angst for the pre-post surgery samples, and everyone else. And thank you very much. And if you have any questions, you're welcome.
Questions for you now? Okay.
Thank you.
Thanks very much.